For a long time, the story of the first humans was told with stones and bones. We had fossil skulls, stone tools, and the layers of dirt they were found in. From these, we built a timeline. It was a story of species replacing each other. It was a neat linear progression. We thought we had the basic picture. Homo habilis gave way to Homo erectus. Then came the Neanderthals in Europe, and finally us, Homo sapiens, marching out of Africa to replace everyone else. It was a simple story, and it was mostly wrong. The tool that shattered this simple story wasn't a trowel or a brush. It was a DNA sequencer. The ability to read ancient DNA, or aDNA, has completely changed our understanding of our own past. It's like we were watching a silent black and white film of human history, and suddenly someone turned on the sound and color. The old characters are still there, but now we can hear them talk. We see their relationships, their families, their health problems, and their journeys in detail we never thought possible. Getting DNA from ancient bones is incredibly hard. DNA is a fragile molecule. As soon as an organism dies, its DNA starts to break down. It's attacked by water, by microbes in the soil, by oxygen. Over thousands of years, it shatters into millions of tiny, degraded pieces. What's left is a mess. It's also contaminated. The vast majority of DNA you find on an ancient bone comes from bacteria, from the archaeologist who dug it up, from the lab technician who handles it. Finding the authentic ancient human DNA is like trying to find a few specific grains of sand on a very large, very dirty beach. For decades, people thought it was impossible to get a full genome from a truly ancient human. But a few persistent scientists, led by people like Svante Pabo, figured it out. They developed new methods, they worked in ultra-clean labs, wearing full body suits to avoid contaminating the samples. They used powerful computers to piece together the millions of tiny broken DNA fragments, like reconstructing a book that has been put through a paper shredder. And in 2010, they did it. They published the first draft of the Neanderthal genome. That's when the old, simple story of our past really began to fall apart. The Neanderthals were our closest extinct relatives. For over a century, we saw them as brutish cavemen, the us that we, the smart and successful Homo sapiens, had replaced. The fossils told us they lived in Europe and Western Asia for hundreds of thousands of years. They were tough, adapted to the cold of the ice ages. They had large brains, made complex tools, and buried their dead. Then, around 40,000 years ago, they vanished, just as modern humans were spreading across Europe. The assumption was simple: we outcompeted them. Maybe we were smarter, better organized, or just more numerous. The ancient DNA told a different story. It wasn't about replacement, it was about mixing. When the Neanderthal genome was compared to the genomes of people living today, the scientists found something shocking. Every person with ancestry outside of Africa carries a small amount of Neanderthal DNA. Usually, it's around 1 to 2%. This could only mean one thing. When our Homo sapiens ancestors left Africa and moved into the Middle East and Europe, they met the Neanderthals who were already living there. And they had children together. This wasn't a rare, one-off event. It happened multiple times in different places. We now know that the story of human expansion wasn't a story of conquest, but a story of contact and connection. These weren't separate, warring species. They were just different groups of people. They saw each other as people. They were close enough biologically that they could produce healthy, fertile offspring. Those children were raised in human communities, and they passed their mixed heritage down through the generations. We are the result. That 2% might not sound like much, but when you add it up across the billions of non-African people on the planet, a huge amount of the Neanderthal genome still exists, scattered in little pieces inside us. But what did we get from them? This genetic inheritance wasn't just random junk. Some of it was useful. The Neanderthals had lived in Europe and Asia for a very long time. They were well adapted to the local climates, foods, and pathogens. Our ancestors, fresh out of Africa, were not. Interbreeding with Neanderthals was a kind of evolutionary shortcut. It gave our ancestors a quick dose of helpful genes. DNA analysis shows that many of the Neanderthal genes that are common in modern humans are related to our immune system. They are genes that help our bodies recognize and fight off certain viruses and bacteria. This makes perfect sense. When Homo sapiens arrived in Eurasia, they would have encountered a whole new set of diseases. The Neanderthals had been dealing with those diseases for millennia and had built up genetic defenses. By mixing with them, we picked up some of that immunity. Other Neanderthal genes seem to have influenced our skin and hair. Some variants are linked to lighter skin tones and different hair types, which may have been advantageous in the lower light conditions of northern latitudes. Lighter skin, for example, is better at producing vitamin D from sunlight, a crucial adaptation for surviving in places with long, dark winters. We even got genes related to our response to things like sun exposure and our sleep-wake cycles. It's a very intimate legacy. The way your body fights a cold, the way your skin tans, or the type of hair on your head could be, in a small way, a gift from a Neanderthal ancestor who lived 50,000 years ago. Of course, not all the mixing was beneficial. Scientists have also found large deserts in our genome, sections where there is no Neanderthal DNA at all. This suggests that some Neanderthal genes were harmful to the hybrid offspring. For instance, there seems to be a lack of Neanderthal DNA on the X chromosome and in genes related to male fertility. This might mean that male hybrids were less fertile than female hybrids, a common pattern when two distinct populations interbreed. Over generations, natural selection would have weeded out the Neanderthal variants that caused problems for us. What we are left with today is the stuff that was either helpful or at least not harmful. The Neanderthal story was a revolution, but ancient DNA had an even bigger surprise in store. In 2008, archaeologists were working in a cave called Denisova in the Altai Mountains of Siberia. They found a few tiny bone fragments. One was a piece of a pinky finger from a young girl. It was so small and insignificant that it could easily have been lost or ignored. But it was sent for DNA analysis. No one knew what to expect. Maybe it was a Neanderthal. They were known to live in that area. Maybe it was an early modern human. When the results came back, it was neither. The DNA was from a completely new group of ancient humans, one that was distinct from both Neanderthals and modern humans. They were named the Denisovans, after the cave where they were found. This was a landmark moment. For the first time, an entire group of extinct humans was discovered not through a large fossil skull or a skeleton, but through the DNA in a tiny piece of bone. Without DNA, we would never have known they existed. The Denisovans were a ghost population, and genetics had brought them into the light. Further analysis of the Denisovan genome showed that they were a sister group to the Neanderthals. They shared a common ancestor that had split off from our own lineage hundreds of thousands of years ago. After that split, the Neanderthal lineage went one way, mostly into Europe, and the Denisovan lineage went another, mostly into Asia. And just like with the Neanderthals, the story included mixing. When scientists compared the Denisovan genome to modern human populations, they found a clear signal. But this time, it wasn't spread all over the world. The Denisovan genetic legacy was found almost exclusively in people from Oceania, especially Papua New Guinea, and to a lesser extent in East and South Asia. Some populations in Papua New Guinea have up to 5% Denisovan DNA in their genomes. This was a stunning discovery. It meant that as our ancestors spread across Asia and out towards Australia, they met and mixed with the Denisovans. This completely redrew the map of the ancient world. It wasn't just a simple encounter between sapiens and Neanderthals in the West. There was a whole other story playing out in the East, with a different group of archaic humans. The world of the late Pleistocene was a much more diverse place than we ever imagined. It was a world with at least 3, and probably more, different kinds of humans living at the same time. The legacy of the Denisovans is just as fascinating as that of the Neanderthals. One of the most remarkable examples comes from Tibet. People living on the Tibetan Plateau have a remarkable ability to thrive in the thin, low-oxygen air at high altitudes. For years, scientists searched for the genetic basis of this adaptation. They found it in a gene called EPAS1. The Tibetan variant of this gene helps their bodies use oxygen more efficiently and prevents the dangerous side effects that affect most people at high elevation. When researchers checked the Denisovan genome, they found the answer. The Tibetan EPAS1 gene was a nearly perfect match for the one found in the Denisovan girl from the Siberian cave. The ancestors of modern Tibetans got this life-saving gene by interbreeding with Denisovans. The Denisovans, who had lived in Asia for a very long time, had already adapted to places like the Tibetan Plateau. When modern humans arrived, they inherited this ready-made solution through interbreeding. It's examples of a functional trait passed from an archaic human to us, something that allowed a modern population to conquer one of the most extreme environments on Earth. So, the picture of our past is no longer a simple family trait. It's a tangled, braided stream. Our own species, Homo sapiens, emerged in Africa, but when groups of our ancestors moved out of Africa, they entered a world already populated by their distant cousins. They didn't just replace them, they met them, interacted with them, and had children with them. We carry the evidence of those encounters within our own cells today. We are, in a very real sense, hybrids. Our lineage is mostly African Homo sapiens, but with bits and pieces woven in from these other ancient groups. Ancient DNA has also helped us uncover evidence of even more ghost populations groups for which we have no fossils at all, only their genetic echo in the DNA of other ancient or modern people. For example, by analyzing Neanderthal and Denisovan genomes, scientists have found traces of their interbreeding with an even earlier, more mysterious hominin group. This might have been Homo erectus or a related species. It seems that this pattern of mixing and interbreeding goes back deep into our evolutionary history. It might be a fundamental part of what it means to be human. Beyond these epic stories of interspecies romance, ancient DNA is also filling in the details of our own Homo sapiens story. It has refined our understanding of how our species populated the entire globe. The old model was a single, dramatic, out of Africa event. DNA shows it was messier than that. There were likely multiple waves of expansion, some of which may have failed and died out, and others that succeeded. The peopling of Europe is a great example. Ancient DNA from skeletons across Europe, spanning thousands of years, has revealed a dynamic and often brutal history. The first modern humans in Europe were hunter-gatherers. They were the ones who painted the incredible art in caves like Lascaux and Chauvet. For a long time, it was assumed that these people were the direct ancestors of modern Europeans. But ADNA shows this isn't true. Around 9,000 years ago, a new group of people arrived in Europe. They came from Anatolia, modern-day Turkey, and they brought a revolutionary new technology with them, farming. DNA from this period shows a massive population shift. The Anatolian farmers almost completely replaced the original hunter-gatherer populations in many parts of Europe. The hunter-gatherer DNA largely vanished, though some mixing did occur. So, for a few thousand years, Europe was a land of farmers with Middle Eastern ancestry. But the story doesn't end there. Around 5,000 years ago, another massive migration swept into Europe. This time, it was from the steppes north of the Black Sea. These people, known as the Yamnaya, were pastoralists. They herded cattle and sheep, they used horses and wheeled carts, and they brought with them the Indo-European languages that most Europeans speak today. Their DNA shows up all over Europe, from Spain to Scandinavia. In some places, like Britain, they replaced up to 90% of the existing population of farmers. So, the genetic makeup of modern Europeans is a three-part mix: a little bit of the original hunter-gatherer, a large chunk from the Anatolian farmers, and another large chunk from the Yamnaya horsemen of the steppes. This complex history of migration and replacement was completely invisible from the archaeological record alone. It took ancient DNA to reveal it. Similar stories are emerging all over the world. In the Americas, ADNA has helped trace the path of the first people who crossed the Bering Land Bridge from Siberia. It has shown how they paused in a region called Beringia for thousands of years before rapidly spreading south all the way to the tip of South America. It has helped identify distinct founding lineages and has even settled debates about how many waves of migration there were. But perhaps the most personal and human stories from ancient DNA come from looking not at entire populations, but at individuals and families. We are now able to get DNA from multiple individuals buried at the same site. This allows us to reconstruct ancient family trees. In one Neanderthal cave in Siberia, scientists managed to sequence the genomes of a father and his teenage daughter, along with several other relatives who lived together in the same small group around 54,000 years ago. This was an incredible glimpse into the social life of Neanderthals. The DNA from the group showed very low genetic diversity. This means they lived in small, isolated bands of maybe 10 to 20 individuals. This analysis of their Y chromosomes passed from father to son and their mitochondrial DNA passed from mother to child showed another interesting pattern. The Y chromosomes were all very similar, but the mitochondrial DNA was more diverse. This strongly suggests that it was the females who moved between groups to find mates, while the males stayed in the groups they were born in. This is a pattern seen in many human societies today. For the first time, we had solid evidence for the social structure of a Neanderthal family. It made them feel less like a different species and more like a different human culture. Ancient DNA can also tell us about the health of past people. We can find the DNA of pathogens, like the bacteria that causes plague or tuberculosis, inside ancient skeletons. This allows us to trace the history of infectious diseases. We can see when they first emerged, how they evolved, and how they spread with human migrations. We can also look at an ancient person's own genes to see if they were genetically predisposed to certain conditions like heart disease or autoimmune disorders. It allows us to study the evolution of disease and health over millennia. We can even reconstruct what ancient people looked like. We can find the genes that determine skin color, hair color, and eye color. The first hunter-gatherers of Europe, for example, had a surprising combination of features: dark skin and blue eyes. The farmers who replaced them brought genes for lighter skin. This is another detail that brings these long vanished people to life, making them more real and relatable. We can imagine them not as abstract concepts, but as living, breathing individuals. The field of ancient DNA is still young and it is moving incredibly fast. Every year, the techniques get better. We can now get DNA from samples that were once considered impossible. Scientists are even learning how to extract human DNA from the dirt on a cave floor, even when no bones are present. This opens up the possibility of finding out who lived at a site without ever finding a fossil. We are pushing the timeline back, trying to get DNA from older and older hominins, getting closer to the great split between our lineage and the lineages of our ancient cousins. What ancient DNA has given us, more than anything else, is a new perspective on ourselves. It has shown us that our history is not a simple story of progress and replacement. It is a story of connection, mixture, in a world that was for a very long time filled with many different ways of being human. It has erased the firm line we used to draw between us and them. The Neanderthals and the Denisovans are not just extinct species. They are part of our story, they are ancestors. Their genetic legacy is written into our own biology, influencing our health, our appearance, and our ability to adapt to the world. The simple story from Stones and Bones was easy to understand, but it was incomplete. The story from ancient DNA is complicated, messy, and constantly changing as we learn more. But it feels more true. It shows that our species is not the endpoint of a straight, triumphant line, but the last surviving branch of a rich and diverse family tree. It would be more incredibly expensive. The story is in our bones, but the details, the secrets are in our DNA, and we are only just beginning to learn how to read them.